I'm going to get rid of that. I do actually know what that's showing, but it's more than we need to know right now. So here is, I actually don't remember where I got this. This is a smaller image and it's kind of grainy, so it's a little bit hard to see, but you can see DNA polymerase is bound to both strands at the same time. And then there's this little like complex in here that's got the DNA helicase. This image shows them sort of attached to each other, but they're kind of one behind the other. And then you've got your polymerase. This one doesn't really show them connected in the middle, but they are connected down below. All right. So our polymerases, I think you probably have most of this already. This is just sort of general function of the DNA polymerases. Going to use the template strand to add in nucleotides from the 5' to 3' direction. We're using our deoxynucleotide triphosphates to catalyze that reaction and also add in the nucleotides, and then we need that primer available. So RNA polymerase is going to come in first, lay down that primer, and then DNA polymerase is going to come in behind and add in after that. So in E. coli, there are two major DNA polymerases. DNA polymerase 3, which does the bulk of the synthesis, and then polymerase 1 is going to remove those primers. In eukaryotic cells, there's actually a whole slew of other DNA polymerases that do all kinds of like proofreading activities and things like that. So there's quite a few more in eukaryotes. In prokaryotes, they kind of have limited proofreading activity, so the DNA polymerases can, to some extent, correct when mutations happen. Eukaryotic cells are much better at it and do many more DNA repair options, which really just means that mutations accumulate in prokaryotes a little bit faster than they accumulate in eukaryotes. All right. So again, this is just kind of summary to make sure we've got it. We have our polymerase. I know we already wrote this down. Catalyze the formation of that phosphodiester bond at the 3' end, and then we've got our newly synthesized strands that are complementary and anti-parallel. So this image sort of shows what's happening with DNA polymerase. In a minute, I'm going to draw out what's going on with the replication bubble, and I'm going to leave out a lot of this stuff. And then I have another video to show you that shows DNA polymerase in all its messiness. So basically, we've got the polymerase that's on the leading strand. That's just going to kind of move directly, continuously towards the replication fork. And then in order for this top half, which is moving one direction, and the bottom half to also move in the same direction because they're connected, the DNA strand has to loop around and be fed through sort of in the opposite direction that it is running. So we get this little loop at the end on one side so that the polymerase can move in the same direction on both strands, even though, like, physically move in the same direction, even though the lagging strand is actually being made in the opposite direction. So DNA polymerase polymerase 3 does have what we call exonuclease ability. So exo means to remove. And then the nuclei part, that's for nucleic acid. So exonuclease can remove a nucleotide if DNA polymerase recognizes that it puts in the wrong nucleotide. It can actually pause, take it out, or excise it. and then put in the correct one. So I think sometime last week, maybe Mia, you asked a question about base pairing and if they could, if your nucleotides could pair to the wrong thing. So what happens here is that if DNA polymerase recognizes, because remember we talked about a little bump or a little pinch in the structure, if it recognizes that that's happened, it can actually stop, back up, pull out the nucleotide and put in the right one.
Okay.
So it can kind of do that as it's going.
Okay.
All right, so I'm going to draw out DNA replication. So I'm going to start, we're going to sort of jump in at the place where DNA helicase is at the replication fork. So I'm going to draw it and then we'll see. I might leave it, might not. DNA helicase binds and is going to move along the replication fork and open up the replication fork as it moves. So DNA helicase opens the replication fork. So we've already got those initiator proteins that have come in, opened up the bubble. I'm not going to draw them, but we have single-stranded binding proteins on our molecule to help keep that open. And then up ahead, I'm going to draw it way out here. We've got DNA gyrase that is moving ahead of the helicase to make sure that we're undoing the twisting that's happening on our molecule. So I am going to label the strands. I'm going to make the top one the 5' strand, the bottom one 3' on this end. So these guys are anti-parallel. So over here, we've got the 5' end. You should, for the next exam, be able to identify the 5' and 3' ends. So there's a couple ways we can do this. We can know which ones are our 5' and our 3', and then identify which one is going to be the leading and the lagging strand. Or we can identify which one's it's leading or lagging, and then place the positions. So you should be able to recognize which end is which on all of your strands. You can always keep track of that because DNA polymerase builds in that 5' to 3' direction. So if you can figure out which way your strand is being built, you can work back from your new fragment to figure out where the polarity of your strands. All right, so we'll say that our origin of replication is up here. That's our origin of replication. And then over there on the other side, we've got the other half of our replication bubble. So we're going to make like a ghost replication bubble, because we've got one on that side as well, but that's too much to keep track of at the moment. So our first thing, what's our first enzyme that needs to come in? What do we need to do first? After we've got helicase. Teresa? Okay, so helicase comes in. Helicase is going to open up that replication fork. Which one comes in next? Primase, very good. Okay, so primase is going to come in. Primase is going to bind on both strands. So I'm going to say like step 1A, step 1B. We've got our helicase and our gyrase. Step 2, primase adds a primer to both strands. So I'm going to draw DNA polymerase as sort of two separate enzymes. We'll keep in mind that it's actually one, but I am not artistic enough to draw it all together and draw the loops and all that business. So primase is going to come in. What color do we want primase to be? Pink. Oh, you don't get pink. Sorry.
Green.
What did you say? Green. All right, we'll do light green on primase. Okay, so primase is going to come in, and it is going to lay down a little track of RNA. So this is going to be our RNA primase and primer. And we are going to build this strand, the opposite, 5' to 3'. So our top one in this image is going to be our leading strand. And the only reason that the top one is the leading strand is this image is because when I started teaching, the textbook had the leading strand on the top, and I just always draw it that way. Does not have to be. The top one on this side of the bubble could be the lagging strand. But the reason that we know that this is leading is that we've got 5' to 3' towards that replication fork that we are zoomed in on. So we're going to build 5' to 3' going that direction. So RNA primase is going to come in, put in that primer. The next, this is also happening on the lagging strand as well. So we've also got the lagging strand. Primer is going to come in here, or primase is going to come in here, add a little primer on the lagging strand. So on the top, once DNA polymerase comes in, it's going to bind after the primer, and it's going to work towards the replication fork, and it's just going to keep pushing this replication fork more and more open. On the bottom, we don't really have room at the replication fork to get all the way in there, and so it's going to start a little farther down away. And what's going to happen is as we open this replication fork, we're going to have more single-stranded opened up DNA, and we're going to get more primers that are going to come in as we open. And we're going to build our strand in these little fragments as we open up. So down on the bottom, that's going to be our lagging strand. And this is going to be built away from the replication fork. As helicase and polymerase open more double-stranded DNA. All right, so primase is going to come in, start adding primers, and it's going to, on the lagging strand, continue to add primers as we open up more and more. So then, what color do we want DNA polymerase to be?
Oh, you already have purple.
Red, okay. All right, so now DNA polymerase, our big DNA polymerase molecule is going to come in. It's going to bind, oops, I just put that in the wrong spot. Oh, goody, okay. It's going to bind right there. So this is going to be our DNA polymerase. And keep in mind, these two parts of the molecule are actually connected. We've got some weird twisting, loopiness happening so that it can actually physically move in the same direction. So what's going to happen is DNA polymerase is going to move, adding nucleotides towards this replication fork. And we're going to add nucleotides as we go, again, in that 5' to 3' direction. Let me add that in. Okay. Nope. So DNA polymerase binds both strands at the primers and begins elongation. And then sort of a 3A step, or 3A and 3B, however you want to label it. As the replication fork opens up, new primers are added. And new fragments are made. Yes.
On the leading strand, the second DNA polymerase, is that a separate enzyme or is it like it was there and moved?
Yeah, it was there and it moved. Yeah.
Okay.
So similarly, I'm going to draw one here. I'll do it in a different color, a slightly different red. So once this fragment gets built, it sort of bumps into the next one, then the DNA polymerase allows the single strand, the lagging strand, to sort of thread through the molecule and then binds at a second place. So up on the leading strand, the DNA polymerase just stays bound, and it's kind of like a car on a track, like on a train track. It just keeps going. Whereas this one on the bottom, it's actually going to let go and move down to the next one, and then build another fragment. So this is going to build a fragment here.
And then it hops backwards.
Mm-hmm. And then as we open up even more, it's going to hop backwards again. And on the next slide, I'll kind of zoom out and show you all the little fragments and then the last couple of steps. All right, so I'm going to redraw this on the next slide. What I'm going to draw is sort of a more expanded replication bubble. So we've had our primers come in, we've had DNA polymerase working on our molecule. So on the leading strand, I'm going to draw in our primer and then draw in our newly synthesized leading strand. And so now we've got DNA polymerase here. We've still got helicase there at the replication fork. We've still got topoisomerase up at the front. And then on the bottom, what we have is a series of these fragments that are primers along with the new piece of DNA. So then our next step, step 4, we are going to have another DNA polymerase 1. That first one was called 3. Sorry, I didn't tell you that. But DNA polymerase 1, which is also called exonuclease, is going to remove the RNA primers. So wherever they are, so the one on the leading strand, and then the multiples on the bottom, and replace them with DNA. So then what we have are these little fragments. And they have been replaced with DNA. So I'm going to use my other red just to make a point. They've been replaced with DNA. So we've got all DNA now. But these little fragments are not connected to one another because DNA polymerase can only make that OH to phosphate bond, that 3' OH to 5' phosphate bond. What it cannot do is connect this phosphate over here to this oxygen over here. And so we need one more enzyme. Let's see what colors we have left. Orange. Yep, orange or yellow, but yellow is not going to work too well. So we need one more enzyme. DNA ligase links the fragments. So what it's doing is just forming a bond right here between our existing nucleotides. And so what we end up with, this one too. We end up with a fully DNA nucleotide molecule that has been linked. And so what's happening is that as this one is opening up, if we are on a bacterial chromosome, we've got our replication bubble. And DNA replication is happening from the replication each direction. And it's just going to keep going until we get all the way around the circle to the other side, and our fragments are going to connect. If this were a piece of linear DNA, we would have all of these little replication bubbles, and we would have new pieces of DNA, and they would just continue until they run into the next fragment. So we would get replication happening on all sides until we hit the next one. And basically those bubbles just fuse together until we get all the way across our molecule. On the other side, actually, I'll wait. I'll let you catch up a little bit. On the other side of our replication fork, we can kind of think through what's happening. So we looked at what is the left-hand side. So starting at the origin, we built our DNA molecule. We've got our fragments on the other one. So if we look back at our polarity of our molecule, we've got 5' here, 3' here, and then each one of these fragments are 5' to 3'. So now let's look at what's happening on the other side. On this side, we look at the bottom. Let's go with blue. On the bottom, DNA polymerase is going to come in. And it's going to build 5' to 3'. In this quadrant here, because we are building 5' to 3' towards the replication fork, this opposite quadrant becomes our leading strand. So it's sort of like the mirror image of what's happening up here. We're always building the bottom strand 5' to 3', but when our replication fork is in the way, we have to build our little fragments. But over here, if we start at the origin of replication and build 5' to 3' with another DNA polymerase, we can just go straight in towards that replication fork. Because over there, we also have a helicase. I think helicase was perfect. So we've got another helicase here that's opening up that way. Up on the top side, we've got our other lagging strand. So we can build 5' to 3' away from the replication fork. But as that replication fork opens up on this side, we get our fragments on that end. And so both ends are just going to kind of expand out. The reason that we call it the lagging strand is that replication is forced to wait until the leading strand forces the replication fork open or open farther. So it's not that it's any slower, really. It's that it just kind of gets hindered by waiting on that leading strand. So the actual speed of replication is the same no matter what strand you're on, but that bottom one is always just a little bit behind. Or sorry, the lagging is always a little bit behind because it's sort of waiting for replication to happen. Are there any questions on this process? All right, so overall, DNA replication is pretty accurate. Oops, I'm sorry, Nia, were you finished? I can wait. But there's a couple ways that the cell sort of ensures that mutations don't happen. So DNA replication is not perfect. It's kind of like when you have a paper and you read it, and then you have your roommate read it, maybe you have your parents read it, proofread it, take it to the Success Center, whatever, and then you still end up with a typo. Because sometimes that happens, right? So DNA replication is the same way. There's lots of checks in there to make sure that mutations don't happen. So it's kind of like when you're using your word processor and you see the little red line that says that it's misspelled, and you pause and you autocorrect it, or your text message is autocorrect or whatever, happens right away. So the DNA polymerase can do that. It can recognize as soon as it's put in the wrong one that something has gone in wrong. It can also go back. So just like you might hand your research papers to somebody else to proofread and they catch your problems. Oh wait, we've already got this. I already wrote that down. There we go. DNA polymerase 1 can also go back and act as a proofreader. So polymerase 1 is also our exonuclease that removes the primers and puts in the correct DNA bases in place of RNA. But it can also act as a proofreader. So if the cell recognizes that it has inserted the wrong base, after DNA polymerase 1 goes through, it can still make that correction. So that correction is called mismatch repair. It doesn't catch everything, but it is based on that idea that we talked about last week, where if you put in whoa, I don't know what, that's not what I meant to do. If you put in a purine instead of a pyrimidine, you're going to get, hey, I want my pen back. You're going to get sort of, oh, no, I cannot draw today. Okay. You're going to get bumps in your structure where you have mismatches. And so DNA polymerase 1 actually sort of goes along the strand and makes sure that we don't have any of those unusually paired bases. It can't catch everything. So for example, if you put in an adenine instead of a thymine, a cytosine instead of a thymine, you might actually not have that the same structural problem. Sometimes the nucleotides can also do weird things. There we go. Where the or DNA polymerase can accidentally put in multiples that can also sort of cause a little bump because if it puts in extras, there's nothing for it to be paired with, and so it can also sort of bulge out. So DNA polymerase 1 is really kind of looking for structural problems that make the molecule like bend or twist or do weird things.
What does fidelity mean?
Fidelity means like accurate, like ensuring that it is like correct. It's like another like a synonym for accuracy. All right. Are there any questions on the process leading strand, lagging strand fragments? These guys are the Okazaki fragments.
So what enzyme is making those RNA bases?
Putting those in. The primase is actually an RNA polymerase. Or you mean which one is building the nucleotides?
I guess both.
Because he said making the bases.
Like what enzyme is putting it into the replication?
Putting the RNA bases in?
Yeah.
That one is the primase, which is an RNA polymerase.
I was gonna ask about the Okazaki.
Oh, yeah, you did. Yeah, Okazaki. Yeah, it's all those little fragments.
You
Yeah.
said RNA polymerase.
Yes. So primase is an RNA polymerase. So polymerase means an enzyme that builds a polymer, and a polymer is a long chain of repeating units. So for example, carbohydrates like starch or cellulose are polymers of glucose, long chains of repeating glucose. Proteins are polymers of amino acids, long chains of amino acids. So DNA and RNA are polymers of nucleotides. So polymerase is just telling you A's means that it's an enzyme. So anything that ends with A's is an enzyme. So if you look at this sheet, you know they're enzymes because they end in A's. But it's just telling you that it is building a polymer of RNA. And then we specifically call it primase because it's a special version that is working with replication. All right. This table, I did not give you this table. This is all of the different polymerases that are involved in eukaryotic cells. And I've actually never studied most of them. My master's degree was in DNA technology, not molecular biology. So I didn't actually ever learn a lot about the other polymerases aside from the common ones that are doing synthesis and the exonuclease, the polymerase 1. But there are specific polymerases that do specific types of repair. So depending on what type of repair or mutation replacement it has to do, there are different types. I really just want you guys to know polymerase 3, which is the big replication polymerase, the synthesis polymerase, and then exonuclease or DNA polymerase 1 that is performing the primer excision and the proofreading. So you don't have to know all of these other ones in eukaryotic cells, but there are a whole bunch more. And then we also have DNA replication happening in the mitochondria and the chloroplasts. So those guys have their own set of polymerases. So for example, the gamma polymerase, that is the one that's found in mitochondrial DNA. So it's specifically working in the mitochondria. All right, so eukaryotic replication is essentially the same as bacteria replication, except that we've got that linear chromosome with lots of origins, and then we've got more repair and mutation correction options with other polymerases in eukaryotic cells. But there are two other things that make replication a little more complicated in eukaryotic cells. One is the presence of histones. Bacterial cells don't have histones. Remember, histones are those proteins that are built of 8 subunits that DNA wraps around to help sort of keep our supercoiling in nice, neat order. So we have to deal with histones. And then we also have to deal with the ends of the chromosomes, the telomeres. So in bacteria, we don't have telomeres because they're circular. So telomeres present a special problem for replication. So as far as eukaryotic, we have to unwind the DNA from the histones. So we have to do that first before DNA replication can take place. And then after DNA replication, the histones come back in and wrap that DNA up right away. So we don't leave it sort of disassembled and pulled apart very long. The cell wants to compact it really quickly because this is all happening in preparation for cell division. But that's just a short part of the cell's life cycle. So the rest of the cell's life cycle, it's doing its job. It's expressing the genes that it needs for its job. So we need to get the DNA back to functional relatively quickly. Cell division needs to occur relatively quickly so the cell can actually do its job in the organism. All right, so then we have the telomeres. So at the end of our linear chromosomes, what's going to happen is that we are going to get to a point where in our replication bubble on the leading end, so let's say that the bottom one is our leading strand. On that leading end, we are going to get to a point where DNA polymerase is bound. And DNA polymerase is eventually going to run off the end. But what happens is at the end, DNA polymerase can't replicate the very end. simply because it is bound to the end, and so it can't actually replicate all the way out to the end because it starts to fall off. So what happens is that at the end of every single one of our chromosomes, we're going to have a really small portion that ends up as single-stranded only. So the other side is going to have been replicated from the beginning, but on the leading strand, it's going to stop. And we're going to have this small portion that doesn't get replicated. So what happens is that at the end of the chromosome, every time you replicate it, you lose a little bit because the cell sees this single-stranded DNA and then it degrades it. Because DNA is not supposed to be single-stranded, so essentially we lose just a little bit every single time that chromosome replicates. Hold on, did I draw that backwards? Maybe it's actually, hold on. I've now confused myself if it's leading or lagging. And I've gone to the wrong end. I need a rewind and a do-over. All right, rewind. Let's start this slide over, sorry. Okay, same problem though. Okay, so let's back this up a little bit. I'm going to draw it down here way longer. Okay, on one of our strands, we've got a primer on this side. And that primer is going to cause the DNA to be replicated in this direction. As I was saying that DNA polymerase falls off the end, I was thinking that I was making things up. And I think that I was. Okay, so the problem is not that the DNA polymerase can't go all the way to the end. The problem is that on the other end, we have a primer. And on that other end, we cannot get, I wasn't totally wrong, we cannot get our exonuclease in there to replace that primer that is on the end. So when this strand replicates, and then our exonuclease degrades that primer, we don't have a 3' end on this side that we can add in our remaining DNA to. Because remember when we were over here on our Okazaki fragments and we had a little primer, we had a primer there, we had a 3' end that we could link it to. But at the end of our telomeres, we don't have a 3' OH over here that we can replace that primer or replace that primer sequence with DNA. And so what happens, not that DNA polymerase falls off the end, totally made that up just now. What happens is that primer gets degraded and you end up with a fragment without the primer that is just single-stranded on the very end. And so that single-stranded region can get degraded. So at the ends of some of our chromosomes, we have structures called telomeres. So these telomeres are long sections of basically gibberish DNA. They are repeating sequences that are there so that when this region gets degraded, because the primer was there, you don't actually lose any important information. And so in most of our cells, what happens is the end just kind of gets a little shorter every single time. But it's okay because we've got this cushion sequence, the telomeres, this cushion sequence that is allowing the cell to have this little buffer where it can lose sections of this non-coding sequence before we get into the important DNA. So in some of our cells, though, our cells that are dividing very rapidly, like our early embryonic cells, our stem cells, these cells that are going to be performing replication over and over and over again for cell division, some of them will actually express a special type of DNA polymerase called telomerase. And so what telomerase does is actually extends the ends of our chromosomes in some cell types. So what happens at the telomeres in the cells that express telomerase is that we get a little RNA molecule that is complementary to that repeating telomere sequence at the end of our chromosomes. And so what it does is it comes in, binds to the end of the chromosome that would be lost because that's where the primer is. And then telomerase acts as a little DNA polymerase and extends the end of the chromosome out through that RNA polymerase region. Sorry, RNA sequence region. So this little green molecule here, you can see this is complementary to a small region of the end of our chromosome. And so what's going to happen is that our nucleotides get added to this end of our telomere just to extend it a little bit. And that allows DNA polymerase to come in and extend the original part of the chromosome just a little bit. So for telomerase, what it's doing is using an RNA template to extend out that little piece of DNA. I thought you had a question.
Okay, so this is, well, I saw on the cheat that the degrading of the telomeres cause aging and stuff. So is it, and the DNA replication that causes this happens from birth because it does that and stuff. So is it just that like it only starts to take off the important DNA once we get older, or?
So I think that's actually the very next slide. Oh, I'm going to get to that in just a minute.
Okay.
Yes, essentially, in mice. All right, but there's one slide in between. Okay, so not all cells have telomeres. Only some of our cells do. So like, for example, your skin cells don't have telomeres. Your epithelial cells inside all of your organs don't have telomeres. Single-celled eukaryotes have telomeres. I'm not entirely sure why they do, but we do not. Are your early embryonic cells, because those are dividing really, really fast, and they're dividing a whole bunch. And then at some point during development, telomerase gets shut off, and most of our cells stop producing it. And then we have some of our cells that reproduce all the time, like our bone marrow cells, some of our intestinal cells that are constantly being shed. Those ones have telomerase. But most of our somatic cells do not have telomerase. So most of the cells in our body don't have telomerase, and instead they're just continuously shortening every time they degrade. I'm sorry, every time they replicate, because that end, that single-stranded end, is degrading. Okay, so to answer Amira's question, I said yes in mice. For a while, there were some studies in mice, and these studies showed that mice have similar telomerase expression to what we do. And there were several studies that showed a link in mice between shortening telomeres and aging. So basically what's happening is that as the cells are losing the ends, there comes a point where it starts to get into important coding DNA. And then when that important coding DNA isn't there, then the cell dies. And so what happens, if you think, like, the way that I like to explain it is, like, as you get older, like your your layer of skin gets a little thinner, and your skin tears more easily. Basically, it's not being replaced at the same rate. Like, your cell division kind of slows down. Not as many cells are dividing. You just don't have as many cells there. So the cell just stops dividing. So basically what's happening is you kind of start to lose your cells. So there's been a bunch of studies some studies in mice that have shown a link between lack of telomerase and aging, but they haven't been able to show it very well in humans. And that could mean one of two things. It could mean that telomerase, there's some other mechanism that is slowing this process, or it could mean they just haven't observed it well in humans. So for a while it was like, oh, telomeres and aging, and then they were kind of backed off a little bit and said, well, maybe not. They've shown it in mice, but they haven't been able to show it well in humans. So I don't know enough about telomeres to know if, like, it makes sense. I feel like it would make sense that it would be happening in humans as well. There's just not a lot of evidence for a direct correlation. I would say generally, yes, it's probably happening though. So some of the signs of normal aging in mice that have been engineered to not have telomerase are graying hair, hair loss, and delayed wound healing, which is kind of a normal aging progression. By the way, don't hit 40 because your hair all turns gray and that's the end of it. Just saying.
I'll try not to.
Actually, my hair started turning gray at like 27. I wasn't even in my 30s yet. My hair was turning gray. So, and then our book does talk a little bit about a study that was done in zebra finches that sort of supports the studies in mice. And that is that during adolescence, birds that had longer telomeres lived longer than birds with shorter telomeres. So that would sort of suggest that there's some link between telomeres and cell aging. One last slide, and that is that one of the common characteristics of cancer cells is that cancer cells often have telomerase flipped on. So we don't really cover cancer in here. The reason that we don't cover cancer in here is that cancer is more of a cell biology problem rather than a genetics problem. So in a genetics class, we're talking about passing on genes to the next generation. In cancer, there are genes that are heritable, but most cancers are, or at least many cancers are environmentally controlled. So we don't actually talk about cancer too much in here. But there are many cancer cells that express telomerase, and one of the characteristics of cancer cells is that they can divide indefinitely. So this would suggest that having telomerase allows cells to continue to divide, which is sort of the opposite of what's happening in most of our cells that don't have telomerase. At some point, they stop dividing and then they die. All right, so are there any questions? I'm going to go back to the telomerase because I dorked that up royally. Are there any questions on telomerase or telomere replication? Okay, I'm going to go back. What?
This is very interesting to think about.
Yeah. Yeah. So Jacob, I lied a little bit. We don't talk about telomerase in 101. You were asking about the difference between this and 101. There's a little bit of a difference. So next week, we have 2 chapters for transcription and translation, and I really want to get back on track. So I think I'm going to cut down chapters 10 and 11 and just do it in 1 week and save a little bit of time because we're going to need, I don't want to push it back because we're going to need time for gene expression, control, and evolution at the end, and we'll run out of that time for evolution if I don't. So I'll probably cut it down and give you kind of a watered down version of transcription translation. So Wednesday, quiz chapter 7 and 8. So 7 was viral DNA and bacteria. Bacterial DNA and viruses. And then 8 was structure of DNA. So we'll do that quiz right at the beginning, and then we will finish up our PCR lab. Cool. All right, I will see you all on Wednesday.